Okay, class, so today we're going to cover translation, protein folding, and proteolysis. The first part is going to be mainly on translation. So last time we covered transcription and transcriptional regulation, and translation is actually the second step in the central dogma, and that's from messenger RNA to protein. And translation is the process that reads the messenger RNA and makes it into a protein, and this actually occurs on the ribosome. So I know some of you guys have probably already had this in great detail, but we're going to really focus on a lot of key steps in today's lecture. And in part B, we're going to actually look at some protein folding and then proteolysis, which can actually remove misfolded proteins or unneeded proteins. So I'm sure you guys have all seen this in, or something similar to it in, your basic biology courses, but the genetic code is actually interpreted in triplets, okay? And here's just basically the triplet sequences, and these are the amino acids that the triplets encode. Now, there's 20 amino acids and 4 nucleotides, right? A, G, C, and U. And so if it was just 2 base pairs long, it could only accommodate 16 amino acids, right? 4 by 4. With 3 base pairs, you can actually accommodate 64 different codons. So some of these codons are actually used multiple times. And if you'll notice, a lot of them sort of vary in the 3rd position. There are instances where they vary in other positions as well, but in most instances, the first 2 are the same, and then they sort of can vary in the 3rd position, like you can see for proline and valine, threonine, glycine, and alanine. And then some of the others are just used a couple of times. Now, codons are actually read in a specific reading frame from 5' to 3'. Okay, so, and depending on the frame that's actually being read, that's going to change the amino acid sequence. So this is reading in frame 1. You're going to have leucine, serine, valine, and threonine. If you're in frame 2, that same exact messenger RNA sequence is going to be serine, alanine, leucine, and proline. And then if it's in the 3rd, glycine, arginine, tyrosine, and histidine. Now, one of the key elements in protein synthesis is actually the transfer RNA, or tRNAs. Okay, and the transfer RNAs are what actually bring the amino acid into the ribosome so that it can be added to a growing polypeptide. Here's just a simple sort of schematic of a tRNA with its secondary structure. Here's the primary sequence of it, and you can see there's a lot of hydrogen bonding in the secondary structure. And that actually folds into a tertiary structure that looks something like this. And you can see all the hydrogen bondings. You'll also note in the tRNA that a lot of the nucleotides are actually modified, okay? And so some of them are like the D-nucleotide here, which is dihydrouridine, and the Greek psi here is pseudouridine. So these can actually be modified, and it's sort of good way to think of it is this sort of cloverleaf structure here. And then the amino acid is actually attached to the 3' end. And then you have your anticodon, your D loop, and your T loop. Just an interesting note, this pseudouridine, these modifications to RNA, they're actually what's used in the Moderna vaccine for coronavirus. I think it's N-methyl pseudouridine is actually put in the messenger RNA of the vaccine to encode for the spike protein. And it seems to stabilize it and evade our immune system. So modified nucleotides in RNA have a lot of different uses and functions, right? And we saw them before with the SNO RNAs and with the ribosomal RNAs. Okay, so In order for the proper amino acid to get added to a growing polypeptide, you need a codon-anticodon, and I'm sure you guys have all had this too at some point in time. And it turns out that one tRNA can bind to actually multiple different codons, and multiple different tRNAs can have the same amino acid attached, right? So if you go back to this chart right here, multiple different tRNAs with different sequences are going to have the same amino acid, okay? And one tRNA can bind to multiple different codons in the messenger RNA. And part of that is because of this wobble position, okay? The third position can accommodate a mismatch. And so if you have a wobble base of U, possible anticodons are A, G, or I, inosine, and likewise C, okay? And that's in bacteria. In eukaryotes, it's pretty similar for the most part. Okay, so this is considered the wobble position. So it turns out that even though we have the potential for 64, well, technically 61 different codon-anticodon bases, there's only actually 48 in humans and 31 different tRNAs in bacteria. So not all are used. So this sort of becomes important. Now, once again, there's other nucleotide modifications in tRNAs. Here's a whole bunch of them. NN-dimethyl guanine dihydrouridine. There's 4-thiouridine, where you have the sulfur here, and inosine, which is a deamination of adenosine. So you don't necessarily have to memorize all these, but just know that they're there and know that they're modified. Okay. Now, some of the tRNA sequences are actually spliced. Okay. But the splicing here is different than the splicing that occurs in a messenger RNA transcript. And so this is actually done just by a single protein, and it's cleaved and a section is lopped out. Now, one of the critical steps for tRNAs is actually adding on the proper amino acid. Okay, and so here's the chemistry of the amino acid addition to a tRNA, making it an aminoacyl-tRNA. Okay, so first you have your amino acid, and then the amino acid actually becomes adenylated. So ATP gets hydrolyzed, and then the AMP is actually added to it, to the carboxyl terminal end. Okay, and then this actually gets added to the OH group on the 3' end of the tRNA. So that it's added, and then you have your aminoacyl-tRNA, and then AMP is released. And so specific amino acids are added by specific tRNA synthetases, and AMP is used as the carrier. Okay, so there's a whole bunch of aminoacyl-tRNA synthetases. Here's what the aminoacyl-tRNA linkage actually looks like. Here's your aminoacyl-tRNA. And it gets attached to the 3' OH group on the tRNA structure, and then you have your mRNA. So there's 20 different tRNA synthetases in all, probably one for each amino acid, and the ester bond on the amino acid is actually high energy, and that's sort of where the energy for protein synthesis comes in. There's also GTP hydrolysis, which helps the process, as we'll see in a second, during elongation and initiation. Okay, so here's basically how the aminoacyl-tRNA synthetases work. In this case, the example is showing tryptophan here, and there's the amino acid. This is the R group. The tRNA synthetase will select an amino acid based on its R group, and then the amino acid will become adenylated, where ATP is converted to AMP, and the AMP carrier. Then you have your linkage to the 3' OH group on the tRNA, and that creates a high energy bond. And then once the aminoacyl-tRNA is charged, it can actually base pair, and then this amino acid can be added to the growing polypeptide by the ribosome. Okay, so some of the things you need to know about the aminoacyl-tRNA synthetases, they actually select the appropriate amino acid, and they also select the appropriate anticodon. Okay, and we'll see in a second the quality control that goes on here, but just for an important point here, once the amino acid is added to this tRNA, it can be used by the ribosome. So if the wrong amino acid actually gets added to a tRNA, it actually can be incorporated into a protein. And so regardless of what is there, it's the anticodon that then is all of the quality control in the protein in translation. Okay, and it uses the ATP conversion of ATP AMP to ultimately generate this high energy bond. Okay, so like I said, any amino acid that gets added to a tRNA will get added by the ribosome. Okay, and so you have to actually edit the tRNAs to make sure they're carrying the proper amino acid. It's the only quality control in the protein synthesis. Okay, so if you have the wrong amino acid, it will get added, right? There's no quality control after this step. There is, once the protein's made, if it's misfolded, it can be degraded. But in order to get the right amino acids in, the tRNA synthetase actually has proofreading capabilities. Okay, so here's the synthesis site where that's selecting for the appropriate amino acid, and it will get added to the tRNA, and the tRNA is selected by the anticodon sequence. Now, in the event that the wrong amino acid is put on here, it will actually get sort of moved up into the editing site, okay? And then if it actually fits in there, it will be removed, okay? The correct amino acid cannot fit into the editing site, so it can't be removed. Only the incorrect amino acid will enter this site so that it can be removed. And so, just so you know, this isn't foolproof. It makes a mistake about once out of every 40,000 times. So wrong amino acids do get put into proteins on quite a regular basis. And if it causes a problem with the protein can then be degraded after the protein is fully synthesized. Okay, and this sort of compares to DNA polymerase, where if you have the incorrect nucleotide added, it will sort of be removed and then the correct one will be put in. Okay, but all the selection here is done by its binding versus here, occasionally the wrong amino acid can be put in and then that flips up to an editing site. And if it's the wrong amino acid, it will fit in there and it will get removed. The proper amino acid does not fit in. Okay, so here's basically the structure of tRNA in a tRNA synthetase. The ATP is shown here in yellow. This is the tRNA. Here's the anticodon. And then the amino acid site is actually right in here as well. Okay, and it forces the AMP amino acid into the right location for coupling. And in the event that it's not, and it's the wrong amino acid, it will flip up to the editing site. And if it's the correct amino acid, it'll be excluded. And once again, the selection here occurs because you have the anticodon of the tRNA binding. All right, now on to peptide synthesis. So what ends up happening is when you have a charged aminoacyl-tRNA, the peptide bond is actually formed, and then what you have is a peptidyl tRNA that's attached to the growing polypeptide chain. Okay, so an incoming aminoacyl-tRNA will then basically form a peptide bond with the previous one. You're left with and it'll attack from the N-terminal to the C-terminal end of the protein, and then the OH on the spent tRNA is sort of removed, and then you're left with a brand new peptidyl tRNA molecule that's attached in the growing polypeptide. And this actually happens on the ribosome, as we'll see in a second. But this intermediate is called a peptidyl-tRNA molecule, and it's actually contains the growing polypeptide chain. And this all happens on the ribosomes, and so there's two types of ribosomes. There's free ribosomes that are contained in the cytosol, and they're often involved in producing cytosolic proteins. And then you have bound ER, or bound ribosomes attached to the ER, which are involved in producing secreted proteins and proteins that enter the secretory pathway, which we'll cover later on in the term. Okay, so there's just two types. There's free ribosomes, and then there's bound ribosomes that are attached to the rough ER. Here's what the ribosome looks like. In the case with the prokaryotic one, you have a 70S ribosome that's split up into a 50S and 30S, and then they contain ribosomal RNA sequences. And then in eukaryotes, it's an 80S ribosome that's composed of the 60S and the 40S ribosomes, and then they also have ribosomal RNAs predominantly. And then there's also some proteins. So in the case with a eukaryotic ribosome, there's about 49 proteins with the large subunit, and then 33 proteins with the small subunit. And we look at the structure later in a few slides here, you'll see that the ribosome is predominantly RNA, and the proteins only make up a small fraction of it. But ultimately, these are multi-subunit complexes, and for those of you who want a little trivia question, the S in this is actually a Svedberg number, which is actually a sedimentation unit, all right, and named after the scientist who worked out sedimentation gradients and stuff. So here's what the structure of the ribosome looks like. You can see it's sort of shaded dark green for the small ribosomal subunit and light green for the large ribosomal subunit. Here's basically the large, and here is the small subunit. And there's three sites here that you have to know, and we'll see what they do. There's the E site, the P site, and the A site in a ribosome, if you look at this sort of schematic here. And they're shown here in red, sort of brownish and yellow color here. The E site is the exid site, the P site is the peptidyl tRNA site, and the A site is the aminoacyl-tRNA site, so EPA. So you can just remember it as the Environmental Protection Agency sites, or EPA. Okay, that's the order that it goes in. And so the RNA actually in the ribosome is actually bound to the small subunit, and here it is sort of shown in blue here. Now, the way translation works, it's a stepwise process. Okay, and just like with transcription, there's three phases to it. There's an initiation, an elongation, and a termination. And this is translational elongation as it's happening. Okay, so the way it works is sort of a stepwise process. So here you have your peptidyl-tRNA in the P site. And spent tRNA is actually leaving the exit site, and a newly bound tRNA is added into the A site, or the aminoacyl-tRNA site. Okay, then the peptidyl synthetase actually catalyzes the formation of the peptide bond between two amino acids, and now your peptidyl-tRNA is in the A site. The large ribosomal subunit moves forward, and followed by the small ribosomal subunit moving forward, and then you have a vacant A site. A newly bound tRNA, aminoacyl-tRNA comes in, and the exit site, the spent tRNA leaves the exit site, or the E site. Okay, and then basically the peptidyl synthetase will form the peptide bond between 4 and 5. Okay, so it's just a stepwise process. Definitely know and understand how this stepwise process works, and also know how the tRNA is actually charged to form the aminoacyl-tRNA and the proofreading that's involved in that. Okay. Now, this is all aided by elongation factors. Okay, so in bacteria, there's EF2 and EFG, and the way it works is EF2 is a small G protein, okay, and it's bringing in an aminoacyl-tRNA into the A site, okay. Now, there's two proofreading steps that happen. The first one is if there's incorrect base pairing here, the tRNA can preferentially dissociate. If it looks like there's pretty good complementary base pairing here, the GTP then becomes hydrolyzed into GDP. And we've already seen small G proteins like RAS and things when they hydrolyze their GTP to GDP, there's actually sort of a movement in the switch loop. Okay, and then this positions it correctly and it falls off. And once again, if there's not tight binding here between the codon and the anticodon, it can fall out and preferentially dissociate. However, if there's a good match, the peptide bond is formed by peptidyl synthase, and then the actual large ribosomal subunit is moved forward by another G protein, EFG, which brings in a GTP, and then that gets hydrolyzed and it helps sort of move move the large ribosomal subunit. It gets hydrolyzed and then it can fall out, and then the small subunit of the ribosome moves forward, so you have a vacant A site, and then the spent tRNA can actually leave. Okay, so there's a slight pausing here before the peptide bond is formed, so that if it's incorrect, it can actually fall out. Here's what the complementary base pairing looks like between the codon and the anticodon, and it also forms hydrogen bonds with the 16S ribosomal RNA. Okay, so the proofreading is actually accomplished by these three RNA interactions here between the mRNA, the tRNA, and the 16S ribosomal RNA. Now here's what all the RNA in a ribosome looks like, and greater than 90% of all the RNA in a cell is actually ribosomal RNA, and you can see here the network and the sort of the tertiary structure of all of the RNA folded up upon itself. There's different domains. You don't necessarily have to know that, just know that there's different domains. And here's one of the ribosomal proteins, L1. But as we covered last time, the ribosomes are made in the nucleolus, whereas the ribosomal RNA is made when all of the chromosomes with the ribosomal DNA sequences sort of come together. If you look at this tertiary structure, if you map out the secondary structure, you can see all of the hydrogen bonding that occurs in the various domains. And you can see it's just really pretty significant, all of the different hydrogen bonding. If you look just at the large ribosomal subunit, you can really sort of get a feel for how much of it is actually RNA. And if you go back to the other structure that was highlighted in green, you can start to really pick things out too. But in this case, the RNA is all shaded in gray, and the proteins are shaded in sort of this orangish yellow color here. And so just by looking at it, you can really see the major mass is actually the mRNA. And so this large ribosomal subunit, 27 out of the 31 proteins are actually shown. So you really get a good appreciation of how much RNA there truly is. Okay, so as with transcriptional initiation, there's also translational initiation. Okay, and the way this works is pretty simple, and this is one of those things too that I want you guys to know. And so the way it works is you have the small ribosomal subunit, the 40S ribosomal subunit, with a methionine tRNA, or the initiator tRNA, and that's bound by a small G protein EIF2. Okay, and so EIF2 associates with the mTRNA and the small ribosomal subunit, and it finds and is brought to the messenger RNA, and it's helped to be loaded by EIF4G and EIF4E that are sort of bound to the 5' cap and the poly A tail. Okay, and so once it's loaded on, it's going to start to scan. And once it hits an AUG, a start codon, the EIF2 GTP is basically going to hydrolyze its GTP, okay? And then that's going to actually fall off as you have good hydrogen bonding between the tRNA, the mTRNA, and the AUG codon. And so once the GTP is hydrolyzed, there's once again a conformational change in the switch loop, and then the EIF and the other factors dissociate and the large ribosomal subunit comes in, and then you have your aminoacyl-tRNA coming into the A site, and then you have basically the elongation step. Okay. The one thing you have to know is that during the, another thing that you have to know is that during the whole sort of scanning of the messenger RNA, it's actually utilizing ATP to sort of move along. Okay, and once it finds that AUG and gets the proper hydrogen bonding, the EIF2 GTP is hydrolyzed, it falls off, the large subunit comes in, and then you can start to have elongation. Now, in the case with bacteria, they can be polycistronic, right? And we've seen this before where you have the 3 phosphates on the end, there's no cap, and then there's multiple sequences with multiple AUGs, and these have ribosomal binding sites. In bacteria, they're called Shine-Delgarno sequences. In eukaryotes and mammals, the sequence in and around this AUG right here is called a Kozak sequence. Okay, and that's where the sort of the translational initiation begins. Now, the final step is translational termination, and that happens when you hit a stop codon, UAG, UAA, or UGA. No need to necessarily memorize that. I know you guys are required to do that in some of your other classes, and many of you already know them. That's great, but you don't necessarily, if you call it a stop codon for me, that's good enough. So the way termination happens is when you hit one of these stop codons, this protein ERF1 releasing factor comes in. Now, releasing factor is not an RNA, it's actually a protein. And when it hits an AUG and there's no tRNA to come in, the releasing factor actually binds to that, and instead of adding an amino acid, a water molecule gets added to the carboxyl end of the polypeptide, and that releases it from the peptidyl tRNA, and then all of a sudden everything just sort of falls apart. Okay, and termination is stopped and the protein is released. Now, it turns out that ERF1 is a protein, but it actually looks like a tRNA if you sort of look at it. And this is sort of molecular mimicry. So here is a tRNA, and here is the ERF2. And I believe this is the anticodon sequence. So here is where the ERF2 would associate with the stop codons. Now, I mentioned this last time, but it turns out that multiple ribosomes are bound to a single messenger RNA transcript between the association with EIF4E, G, and the poly A binding protein. Okay, and so what you get is the formation of these sort of Rosetta structures where you have multiple ribosomes, or polysomes is what they're called, on a single messenger RNA. Okay, and you can see the length of the growing polypeptide coming out of here as it's sort of going around. And here's a great EM photograph of a polyribosome in a cell. Now, occasionally specific amino acid, a modified amino acid can be put in. Selenocysteine is an example, and so you'll have a selenocysteine tRNA, and then that'll actually put a serine onto the tRNA, and then that gets enzymatically converted to selenocysteine, and then you have a specific selenocysteine translation factor, small g protein. It will get added in if it has the proper signal via sort of a stem loop, and it'll get added in. The GTP will be hydrolyzed, and then you'll actually form the peptide bond by peptidyl synthase, and that gets added in. Occasionally, you can have frame shifting, and this is actually something that's used by viruses to make different proteins from the same small viral RNA. Okay, so RNA viruses have a very, very small genome, but oftentimes they encode a lot of proteins, some of which we still don't know what they do. But you know, here is just an example of viral RNA. And in HIV, there's this protein called GAG, and it will be read in one frame. And then occasionally there'll be a frame shift to make a GAG pol fusion protein, which is also used by the virus. Okay, and so 90% of the time there's no frame shift, but occasionally there is a frame shift producing this GAG pol fusion protein. And so, like I said, viruses use this. And it's not clear how much the coronavirus is using frame shifting, but I'm sure that it does as well. So it turns out that there's a lot of great antibiotics and inhibitors of translation. Here are some of them: tetracycline, streptomycin, hygromycin, spectinomycin, and in the case, there's also chloramphenicol and some other ones. So some of these antibiotics are great at inhibiting translation. Here are some of the other ones. For the graduate students, you guys should basically be familiar with some of these. You'll use them a lot in the laboratory, and you'll also see them in the literature, so they're sort of important to know. And finally, there's sort of quality control in the translation itself, okay? And this is called nonsense-mediated decay. Okay, so we saw if you have a pre-RNA and then that's spliced, what ends up happening with normal splicing, you're left with these exon junction complexes, okay? And then this messenger RNA, once it's capped and has the poly A tail, it'll go out into the cytosol where it's translated. Okay, and if all goes well, there's efficient translation. You find the start codon and the stop codon, and as the ribosome's moving through, it will actually knock the exon junction complexes off, and you just get translation. Now, in the case with abnormal splicing, occasionally the intron is going to have a stop codon. Okay, in this case, it's UAA. And so if that's inadvertently exported out, there won't be a proper exon junction complex. And as the ribosome is moving down, if it hits a stop codon and there's still exon junction complexes remaining on the messenger RNA, these proteins called UPF proteins come in, and this actually triggers the RNA to be degraded because it's making an improper protein, and there's a nonsense mutation present from the intron that was sort of not spliced out. Okay, so this is nonsense-mediated decay. This is something you guys should all probably know as well. It's a useful thing. The graduate students will definitely come across it in their reading, and so it's just a cool bit of science there. Okay, so occasionally too, RNAs will get broken, and cells can handle the broken RNAs via different mechanisms. Oftentimes they're just sort of shipped off to the exosome to be degraded. But occasionally, if a ribosome is translating on that messenger RNA and it breaks, what ends up happening is it stalls and it doesn't know what to do. And so then what you have coming in is a tmRNA and that will add an alanine, and then it'll continue to scan, and the alanine tag will get put on the protein, and that'll form basically an improper protein, and this is sort of a signal for degradation, right? It'll be unfolded and degraded, and then the broken RNA is sort of released, and that gets cleaved up by the exosome. But this helps to prevent just proteins or peptidyl-tRNAs from sort of floating around. And as anyone who's worked with RNA will know, that even though even though the phosphodiester bond is pretty stable, they tend to break a lot, right? And we've seen that with DNA, and there's no way to necessarily fix it. So this, if it's a single-stranded messenger RNA, and so this is the way that the cell actually handles it. Okay, it makes this polypeptide with an alanine tag on it that basically tells it to be degraded. Okay, so that's it for part A. I'll come back and we'll be talking about protein folding, protein degradation, and how that all happens in just a bit.